now. So it's my great pleasure to introduce Salvador Dura-Bernal, who is a tenure track assistant prof of physiology and pharmacology at State University of New York, downstate, and also a research scientist at the Nathan Klein Institute. So his research uses large-scale biophysical models, as you can tell, and really detailed simulations to understand cortical circuits, but also really thinking about how to integrate data across scales from molecular to cellular to the systems level. So very relevant for really, I think, a great fit for this seminar series. He's also the developer of NetPine, which several of you are probably using. And yeah, is a widely used software platform for multi-scale brain modeling. I saw a shorter version of this talk recently at an event we were at, and was really, really impressed by all the work. So I'm looking forward to digging a bit deeper today. And without further ado, Salva.
Great. Thanks so much. Thanks so much, Petra, for the invitation. I'm happy to say I got tenure this year, so a big update here in my title. And yeah, very excited to share some of our work. So I'm gonna start giving an overview of the sort of multi-scale mechanistic modeling approach we do and how we use these to understand brain function and disease and disorder in general. I'll use one of the models we've used mostly from motor cortex. And then I'm going to go over some of the research projects we have looking at different disease and disorders and potential interventions and treatments. I'll give 4 or 5 examples, and I'm going to finish just giving a quick overview of the software tool that we've used to develop all these models. So yeah, so I'm going to start with how can this multi-scale modeling approach help us to understand the brain. And I want to start by saying or arguing why the brain is so complicated and one of the reasons we think the brain is so complicated. And so I'm going to try to play this video. Let me see if this works. Okay. So one of the reasons we think the brain is so complicated is that it has many different scales that all interact with each other. Here we're seeing a whole human brain, and we can see the gray matter on the outside with representing many different cortical regions. And so all of these are highly interconnected through the white matter tracts in the middle, forming this massively complex macro scale organization of the brain, where different cortical areas, for example, are connected to other cortical areas. So if we zoom in one of these cortical areas, say the visual cortex or the hippocampus or one of these more mesoscale regions, what we see is a highly complex structure as well. So this representation here in the video is not very realistic because actually you would see a much more packed, densely packed set of neurons. Basically, there's only 15-20% space in between the neurons, so it's really tightly packed, and you have a really well-structured organization where you have all these 6 different layers, for example, in cortex, 6 different layers with different types of neurons and very specific connectivity patterns between these different layers. So, layer 2-3 to layer 5 is very strong and depends on the cell type and so on. So at this mesoscale level, you also have this very complex organization in the brain. And then if we zoom in again and go into a single neuron or a single synapse at this molecular scale level or cellular level, you also would see a really complicated structure. So you see, the interaction between 2 neurons in the synapse level. You see neuromodulators, neurotransmitters, you see calcium, the synaptic vesicles. There's a really complex molecular machinery. And so the complicated things that all of these levels, are interacting with each other. So what happens at this molecular level affects the firing of the neurons, which in turn affects the firing of the circuits and the whole brain dynamics. And that goes in both directions. So essentially what I'm trying to argue here is to understand the brain, we need to understand how all these different scales and levels interact with each other and influence each other. Enter full screen. Okay. So luckily for us, I think we are in a very lucky time for neuroscientists because we have an unprecedented amount of experimental data. I'm showing here an example. This is an electron microscopy reconstruction from the mouse cortex. It's 1 millimeter cubed and contains approximately 200,000 neurons and 500 million synapses. And you can see here with the level of resolution that we have access to this data, where you see, a single synapse with really high resolution. And so these data sets can be used to extract very useful information about the anatomy, the connectivity of the circuit, and so on. Similarly, we have a similar data set for the human cortex. So this is 1 millimeter cubed of the human cortex. With around 50,000 neurons, 130 million synapses. In this image, you can actually see this laminar organization I was talking about before with layer 2, 3, 4, 5, 6, and so on, with different types of neurons in different colors. And again, it's pretty surprising to me that we have this data set of the human cortex. So even in this is another image from this data set, and you can see one human cortical neuron with all the 5,000 axons that are projecting to this neuron. So having this type of data at this resolution for human, I think it's really impressive. And so we're seeing, at the anatomical level, we have data at different scales, like I was mentioning, but also at the physiological or functional level, we have these different scales of measurement. So we can measure activity from the outside of the brain, these EEG signals, and we can keep going inside for more levels of resolution with ECOG, LFP, and even intracellular recordings where you can actually record the voltage of individual neurons. So at physiology level, you also have these multiple scales, and then we can also add now the level of behavior. So many of the experiments we do nowadays relate the behavior of the animal to different physiological measures. And so the question is, how can we integrate and put together to interpret all this data from different modalities, different scales, to make sense of what we're recording? And so our suggestion is that using these multi-scale circuit computational models, we can integrate all this data and make predictions that can help to generate testable hypotheses and better understand or provide explanations of what we're recording. So give me one second. So for example, the models can incorporate all the anatomy of the different neurons. These are built into these very detailed models. They can incorporate all the structure of the different layers. They can incorporate molecular level details. And they can actually be used to reproduce these physiological measures at different scales. So you can generate the voltage traces, the LFP, the EEG in pretty realistic ways. And now we're working on ways to incorporate behavior, to link it to behavior, either by direct actuators or through some proxy, like these low dimensional neural manifolds. So the basic idea is here we build these models, and first we have to validate them. So we have to make sure that they actually reproduce the biology and the real behavior that we're observing in the brain, because otherwise they're no good. And once we have validated them, we can start making predictions that can be tested experimentally and then feed back into the model to potentially refine the model and so on. So this is always work that we do very closely with experimentalists and clinicians. So I just wanted to point out this recent review in Journal Neuroscience, where we got together several of the groups around the world who developed these large-scale but very detailed, biophysically detailed models of brain circuits. And just to show that, there's a lot out there already. So there are models of different species from rodent, non-human primates and humans, and different parts of cortex, motor cortex, visual cortex, hippocampus, and so on. So yeah, check it out if you're interested, and maybe one of these models might be useful for your research. So now I want to move on to a particular example, which is this model of motor cortex circuits we've been developing for several years, and I'll use it as an example to illustrate how we build these models and how we can use them. So this is a model of what would be a cortical column in motor cortex, even though cortical columns don't really exist there. It represents around or the neurons that you would find in this piece of tissue with the real densities of neurons. So we're modeling around 300 micrometers by 1.2 millimeters depth. And there you would find around 10,000 neurons with around 30 million connections. So here we incorporated a lot of the data that is known about this circuit, and as I will show now. So to give you an idea, for each of the cell models we included in this circuit model, we reproduced the morphology of the neuron. So here's the experiment showing the real neuron, and here the simulation. You can see it's practically identical. And here it's this FI curve stimulation protocol where you provide different current injections and measure the spike in output. And you can see the experiment and simulation also match very closely. So what we did to obtain this is run these optimization protocols in supercomputers. So for maybe like a month or so, you specify the target that you want to get, and you tune parameters of the cells, usually these conductances or distributions of the different ionic channels within experimental constraints to find the right solution. And you can see here we even reproduce subthreshold responses and so on. So then we do this for each of the cell types in the circuit. And here is a schematic of how the circuit composition looks. So here on the left, we have the cortical column that we are modeling, the 3D tissue that we're modeling. So it's 300 micrometers by actually 1.3 millimeters. We have the different densities of neurons that you would find in each of the layers, because they're actually not the same. So layer 2-3, for example, has more higher density of neurons. And here we have the layer boundaries from layer 1 to 3, 4, 5A, 5B, and 6. So motor cortex is a bit different. It has a really large layer 5B, smaller layer 4, although still present. And of course, all this is taken from literature. And then we have the composition of the different main cell types that you would find in each of the layers. Here we included 3 types of excitatory cells, this intratelencephalic, the pyramidal tract projecting or corticospinal, and corticothalamic. Each one occupies different locations in the circuit. And we included 2 of the main interneurons, somatostatin and parvalbumin, although in recent versions we have added additional ones like the VIP and the neurogliaform. So, okay, so this is the composition, and then we move to connectivity. And here, this is one of the main tasks for us because it's one of the most complex parts. And so we were lucky to work with all this model we built together with an experimentalist, Gordon Shepherd Jr., who focuses on mouse M1. And so he provided a lot of data that we could put together. So all these are papers from his lab. And so he had information about the long-range inputs that project to M1 from different parts of the brain. So for example, from different parts of thalamus, from secondary motor cortex, from somatosensory cortex. And you can see that each one projects to specific layers. But not only that, they project to specific cell types also within those layers. And so we incorporated all this long-range input connectivity. Secondly, we had information about the local microcircuit connectivity, and here's an example. This is showing a really strong projection from layer 2-3 to layer 5A, 5B. And so we incorporated this, which is again layer specific and cell type specific. And even within layers, we had differences. Because for example, layer 5B is really broad. So we show connectivity really strong at the top, but not so much at the bottom. And finally, we had another scale of connectivity, which is at the dendritic distribution of synapses. And here we see the projections from, for example, layer 2-3 to this corticospinal neuron and how they're distributed along the dendrite. So how are synapses distributed along the dendrite for this particular projection? And this is really important for computation and so on. So we also incorporated this information for each of the projections we had information about. All right, so then we put all that information together and we tried to publish the model, and the reviewer said, okay, now first you have to validate against some data to show me that this is doing something real. So basically had to go back to the starting point, and we contacted this other researcher, Ian Duguid in Edinburgh, who was doing these super cool experiments recording from mouse in vivo intracellularly in motor cortex, which is exactly the type of data that we required. So they were recording from different cell types that we had in the circuit in vivo while the mouse was doing different behaviors. In particular, they were doing this running on a treadmill where they had two behavioral states, where the mouse was in quiet wakefulness and when the mouse was moving in the treadmill. So to simulate the behaviors, we started with the assumption that was given by the experimentalists, which was there were changes in the long-range inputs when the mouse was moving. And the two main changes came from motor thalamus or ventrolateral thalamus to M1 and from the locus ceruleus, which provides these noradrenergic inputs to M1 neurons. So then with those, we were able to simulate the activity of the M1 circuit for the two behavioral states, quiet wakefulness and movement, and for the different cell types. And we were able to compare directly to the recordings from this experimental dataset from Ian Duguid. And so here we show an example of a few of these cell types, layer 2-3 IT cells, layer 5B ITMPT, and so on, and showing that we statistically validated the model, firing rates at least. Here you're seeing the the spiking raster plot with the 10,000 neurons here on the y-axis and 2 seconds of simulation. Different colors represent different cell types. All right, so we had some validation for the firing rates, and then we moved on to validating the local field potentials. So here in the model, we can actually simulate quite accurately, realistically, the local field potentials generated by the neurons at different depths. So here we're comparing the LFP at layer 5. We see the experimental data here at the top in blue and the model data. And we simulated again for the two behavioral states, quiet and movement. So you see clearly, obviously, we're not going to reproduce the exact temporal pattern, but statistical features and in terms of oscillations primarily. And so you can see that we captured the change from quiet to movement, which basically in the quiet state, you see much more slow oscillations in the delta range, and those change to gamma frequency oscillations during movement. So we validated again statistically, and just to say that the model, we also run multiple iterations of the models with different randomization seats, simulating different trials and different animals. So we changed the connectivities within the parameters of the model, and we changed the inputs to the model, again simulating trials and animals so that we have some statistical validity of how to compare the models to data. All right, so next, the reviewer actually suggested this approach, which we hadn't really thought about, but it turned out to be quite interesting, which is looking at these low-dimensional neural manifolds, basically collapsing the 10,000 dimensional activity of the circuit, 10,000 neurons, into 3 dimensions and looking at the dynamics. It's quite common, and it's interesting because it's related to behavior. These low-dimensional neural manifolds have been shown to correlate and predict really well the behavior of animals across species, across tasks. So for example, you can predict the reaching direction of macaques or the mice behavior in a maze and so on. So we did it and we got some interesting results. So here you see the quiet activity and the movement are clearly separated in this low dimensional representation. And the movement showed these cycles, which actually corresponded closely with the gamma oscillations. So it gives some interesting insights. And later I will show how we're using this approach to validate a new version of the model against experimental data. All right, so now we had sort of validated the model against firing rates, LFP, and now we were ready to make some predictions. And so, for example, we looked at one of the LFP oscillations, in particular this delta oscillation, and we're trying to figure out what particular cell types and populations of the model were generating this delta oscillation. And so in the model, we have access to everything in detail, so we can actually decompose this signal into its generating components from different populations. And so we saw that the population that contributed most to this oscillation is the layer 6 IT cells, followed by the layer 5B IT cells. These two signals here. So what we found was interesting, we didn't expect, is that they're almost in antiphase, not fully aligned, but practically antiphase. And so that may explain why the original full LFP signal doesn't show these very clear oscillations, but it's sort of patchy and has this irregular pattern. And that's potentially because different LFP signals from different populations are not fully aligned in phase. And then just another example of a prediction that you could make with the model was looking at this manipulation that the experimentalists did, where they removed the input from the locus coeruleus, which led to a misplaced forepaw, so a change in the behavior in the animal. And this has been associated with Parkinson's, some of the Parkinson's disease movement deficits. And so what we did is try to use the model to explain this in a mechanistic way across scales. And so we saw that removing the inputs from locus coeruleus, what it does is it changes the conductance of HCN channels. So it increases the HCN channel conductance, primarily in the apical tufts of the pyramidal tract neurons. That in turn reduces synaptic integration in these apical tufts, which in turn reduces the firing of these corticospinal neurons, which are basically the ones providing activity to the spinal cord. So this kind of provided a mechanistic hypothesis or explanation for this behavior, linking the molecular level to the cellular, to the circuit, and the behavior. All right. So next I wanted to show this variation of the model that we're working on now for a different task. This is also working with Ian Duguid in Edinburgh, and where we're focusing on these low dimensional manifolds as well to try to relate the model or tune the model to behavior. Because of course one of the limitations we have is we are tuning all these firing rates and LFP and so on, but we want to make sure that the model is actually reproducing the behavior as well, and that is always very tricky. So in this new task, it's center out joystick task. The mouse can move forward or backwards, and they have to reach a target and stay there. So it's like a center out reaching task, usually done in macaques. And he's recording with neuropixels in motor cortex and thalamus. And here we see an example of the recordings and the corresponding joystick trajectories. So the first thing we did is analyze this through these low dimensional methods. In particular, we use this zebra method that is quite popular nowadays. And it basically requires putting in all the neural dynamics information together with the behavioral information, and it generates these low dimensional representations that can then, for example, be used to predict the activity, predict the joystick location, the behavior from the neural data directly. And so we show that learning these models from the data effectively allowed us to decode the joystick location. So the orange is the decoded one, and the blue is the original, directly from the neural data. So we showed it's a useful approach and it's related to behavior, which is what we wanted. And so the next step we are actually working on now is essentially trying to do the same with the model. So this is the neural recordings, and this is a representation of the zebra representation of this recording. And now we're trying to do the same with our full M1 model, trying to obtain these zebra low-dimensional neural manifolds from the model and trying to tune the model to reproduce the manifolds we see experimentally. So this is like adding an additional constraint that we've never had before. We always tune to firing rates, LFP, and so on. And now we're trying to tune to these neural manifolds in order to capture some of the behavior. Of course, this is in addition to all the other stuff. So we're, making it more and more constrained. All right. And yeah, now I wanted to quickly mention before I go into the disease that we have other models apart from this motor cortex. And I wanted to mention this model of macaque auditory thalamocortical circuits, which is in a sense similar to the one I just explained. So it was built in a similar way, but using data from macaques. And it's interesting because it also has this model at the beginning, sort of the lower part of the auditory system, where we can simulate any sound, any arbitrary sound. We pass it through a phenomenological model of cochlea up to inferior colliculus, which generates a very realistic representation of the sound that we can then feed into the cortex, auditory cortex model and thalamus model. So basically, we can provide the same sounds we're using with the macaques, we can provide to the model, and it's processed in a similar way. And of course, again, we've validated this model. So this is some spontaneous activity from the model in experiment and model. We've validated the different oscillations that show up in the model, gamma, theta, delta. Also statistically, here I'm showing just examples, but we see similar oscillatory events in the model and experiment. And one of the things, and here's an example of how this model can generate representations across all the scales that I was mentioning before. So from membrane voltages to spike in activity, LFP and current source density across layers, and even EEG signals. And of course, because all of these emerge from the same biophysical models, you will see similarities, peaks across all of these different types of measures that show up in all of them. And finally, just an example of we're using this model again to try to understand the contribution of different populations to the CSD spectral power. And so we're, like I mentioned before, decomposing the CSD signal into the contribution of different populations to try to understand how these CSD patterns emerge, which is still an unanswered question, the relationship between LFP and CSD and so on. All right, so now moving on to the brain disease and disorders, I'm just gonna give a few examples briefly of some of the work we're doing. So this is a grant that we have from New York State, from the Spinal Cord Injury Board, and it's using this motor cortex model that we just explained. We can generate these neural manifolds, and we can also generate EEG from them. So the question we're asking is whether we can actually decode these neural manifolds from the EEG signal, which presumably the answer is no, but we are trying to figure out if there's something there that could be used. And that, of course, this could be used to improve brain-machine interfaces for spinal cord injury patients. So because we have access to the full model and we can inspect every single aspect of it in detail, it's very easy for us to make this correlation between manifolds and EEG. Another project, this is, we have a collaboration with UC Davis and a grant from Harwell Foundation, basically looking at channelopathies. So these are genetic mutations that happen in different channels, for example, in sodium channels that alter the responses of the neurons. And here I'm showing, for example, this is the NCNA2A mutation that affects then the sodium 1.2 channel. And this is the firing activity for a normal channel, and this is with the knockout. And so the UC Davis guys, they had this single cell model which captures these mutations very well and the response of the single cell. And now we put these mutations in the circuit to really see how those affect the circuit level activity. So this is an example, the healthy control activity, and in blue we're seeing the corticospinal neuron responses. And then what happens when you introduce the mutation? So for example, the activity of these layer 5B corticospinal neurons is reduced. And then, of course, the goal of the project is to come up with potential pharmacological interventions to restore the healthy activity. So here we're trying changes in the, for example, synapses, or you could even try things like neurostimulation. So we're doing this for different channelopathies, and there's lots of different mutations for different channels and so on. And these mutations, the channelopathies, usually result in epilepsy or autism, depending on the type of mutation, as well as other neurodevelopmental disorders. So another project that I'm quite excited about is this grant with Columbia, and it's an NIH grant, and the experimental lab found this really exciting finding that modulating the calcium level of astrocytes reduced the cognitive deficits of early life stress in mice. And so they came up with the theory that astrocytes might be integrating different modulators, the four main different modulators: acetylcholine, norepinephrine, dopamine, and serotonin. And they have receptors for each of these neuromodulators, and so it made a very compelling case. So part of the grant is to build a detailed model of these interactions, and the idea is to combine data from different parts of the model, like I was showing before. So they have this microscopy data with fluorescent labeling for different for the neurons, the astrocytes, and different neuromodulator axons. They have spatial transcriptomics for different cells and astrocytes, and so we can know exactly how they're grouped and their locations and so on. They have local field potential and fiber photometry, so looking at the concentrations of the different neuromodulators and even calcium in the astrocytes. And they also have behavioral data. So again, as I was showing before, the idea is to integrate all these four components into the models to try to explain and provide hypotheses of the observations. And we have, well, this is just a schematic of what we're doing. So we have all this data. We're using it to constrain the model, which includes the molecular level now. And we have interactions between neuron and astrocytes mainly. And so then with the models, we can simulate these signals across scales that we can use to validate. So we are started by building models of the effects of synapses on astrocytes in these very simplified models, and how the calcium levels, for example, are affected when you have synapses in the vicinity. And then moved on to sort of the bidirectional interaction between neurons and astrocytes. So not only how synapses affect the astrocyte, but then how the level of calcium, for example, released by the astrocyte is affecting synaptic transmission and synaptic plasticity. So we started with very simple ball and stick models, and now we're moving to more realistic morphology. So this is a reconstructed morphology of an astrocyte, which are really nice and impressive. And so we have a preliminary simulation of the calcium concentration across all the dendrites, all the processes of this astrocyte. And the idea is then to combine these, thousands of these astrocytes within the motor cortex model with all the neurons as well. It's a bit ambitious. We'll see where we get. And all right, and then another exciting collaboration that we're doing right now. This is with Harvard Medical School, and they have these really exciting results where they are using TMS on Alzheimer's disease patients. They have shown essentially they can slow the cognitive decline and the functional decline almost stop it for 52 weeks. So the patients show, you can see here, this is a measure, I think, of this functional, so how much the patients can function autonomously by themselves. In blue is with the TMS protocol, and in gray if they don't receive anything. So it's really outstanding results. Apparently it's better results than any pharmacological treatment right now. They're already in phase 3 clinical trial. And so they contacted us to try to understand what's going on at this microcircuit level and if they could fine-tune the stimulation protocol somehow to improve the results further. So what we did is adapt one of our models to human cortex, because most of the models we have are either rodent or macaque. So we use one of the datasets I showed at the beginning that contains all of the locations of the different human cortical cells. And we use some morphologies from the Allen Brain Institute that have human cortical morphologies to build a preliminary human cortical model that is shown here. And then we also included the connectivity. So this dataset actually includes, this EM dataset includes connectivity between the different cell types. So we included that in the model for human cortex. And we simulated the TMS stimulation. So again, TMS, you can simulate quite accurately the electrical fields and so on. There are models now developed over many years that can simulate the effect of TMS. So here I'm showing one of these spiking raster plots, and at half point here, we provide the TMS stimulation, and you see how this results in some entrainment of the circuit. So then you can simulate different amplitudes of TMS stimulation, weak versus strong, and we saw different patterns of entrainment and so on. So this is still very preliminary, but we're working to make it a bit more systematic and also validate with the TMS evoked EEG signals that the clinical lab has. So we're trying to simulate those now in the model to validate the responses. Again, with the idea of ultimately optimizing the TMS protocols. And finally, I think it's the last example I have. We're using this auditory cortex model to study schizophrenia. One of the more clear biomarkers that you see, electrophysiological biomarkers you see in schizophrenia, is this 40 hertz auditory steady state response, which is essentially if you provide a 40 hertz sound to healthy patients, you get this very strong response in auditory cortex at 40 hertz. It doesn't happen that much at other frequencies, so it's resonant at 40 hertz. But if you do the same for schizophrenia patients, this response has disappeared. So it's a very clear way of identifying patients and even early stage patients. And so we've simulated this response in the auditory cortex model, essentially by introducing biophysical changes that are known to happen for different cell types and different receptors and so on in schizophrenia. And we can simulate the disappearance of this 40 hertz response. And now we're using the model to gain insights into why this might be happening, and as usual, to potentially identify novel treatments. So we have this healthy circuit model in M1. We build the schizophrenia circuit model validated against experimental datasets in M2, and then M3, the identification of novel treatments. This is a grant under review, so we'll see if it flies. All right, and then just to finish off, I just wanted to quickly mention this tool that we have used to build all these models and that we also developed in the lab. So it's called NetPine, which stands for Networks Using Python and Neuron. So it's a tool to facilitate the development of these models, but also the parallel simulation of the models, which is not always easy, the optimization, parameter optimization, and analysis of these brain circuits, essentially. It uses under the hood, it uses the neuron simulator, the neuron simulation engine. So we work very close with the neuron guys at Yale. And the tool was funded for 6 years by NIH. And now we're trying to find more funding. And so, yeah, the motivation was essentially to try to facilitate the incorporation of this experimental data at multiple scales. So when you build models in Neuron directly, it can be hard to incorporate all this data at the long-range inputs, the dendritic scale, the cellular level, and so on. So NetPine has its own standardized declarative language. It's, again, much more human readable, and it essentially allows you to provide the parameters of the model, the biological parameters of the model you want to build in an easy language that then gets translated into the neuron underlying model and runs the simulations for you, does the analysis, and so on. It also allows you to simulate common measures such as local field potential. So all this is built in. And things like EEG, like I was saying, again, built in using other tools that are connected to NetPine. And an interesting interaction that I was mentioning at the beginning is this interface between NetPine, which is a microcircuit simulation tool, with the virtual brain. This is a whole brain simulation tool that uses these mean field models for each of the brain areas. So what we did in this collaboration with Petra Ritter, is replace one of these nodes that usually would be modeled with a mean field model by one of our detailed microcircuit models in NetPine, and then co-simulate both models at the same time, such that they exchange information. So the activity in the circuit model, the spikes, are converted to signals that can be exchanged between the whole brain model, and then the whole brain mean field activity is converted to spike times that are used as input to this microcircuit model. So this was really exciting for us because, we usually have these models in isolation with relatively non-realistic inputs, and now we could embed it in a whole brain simulation and see the interactions from the whole brain to the microcircuit and from the microcircuit back into the whole brain. So we have also a package that is open source to do this kind of models, and yeah, we welcome anyone who's interested to use it as well. And final estimation, we have this graphical interface for the NetPine tool, which allows you basically to build the models online. You can provide all the parameters across all the different components. You can run the simulations. You can visualize the model and the output of the model. You have all these different measures, including the LFP and spectrograms and concentration of calcium. You can see everything in one screen. We're quite happy that many different labs are using the tool, and many models have been developed for different parts of the brain, different cortical regions, different stimulation protocols, different disease. So there's a large library, and most of these are actually open source and available. So you can take whatever you're interested in and then modify it and use it. And of course, this has led to a number of, I think it's over 30 publications now and multiple grants that use the tool. And yeah, it has been growing over the years, of course. And we are also quite active in dissemination. So these are some examples of courses that we've done around the world. We usually do one every year at the Computational Neuroscience meeting, CNS, which this year is in Halifax, and next year is in Gran Canaria in Spain. And then we've done courses in South America, in Colombia, in Chile, in Argentina, Brazil. So there's many options to learn the tool. And of course, we have the website, netpine.org, which has all the information that you need, the documentation. We have lots of tutorials, including video tutorials on YouTube over 40 hours, lots of example models, a Q&A forum. So check it out if you're interested. And yeah, just to finish off, thank all the funding agencies and all the people that did the work in my lab and other collaborators. And thank you all for listening.